Hello, my name is Madison Sprung, and I'm going to be presenting over the protein rhodopsin today. The 3D structure can be seen on the right here, and it is primarily composed of alpha helices. It is 348 amino acids long. The amino acids that primarily compose the protein are alanine, leucine, phenylalanine, and valine. They occupy the hydrophobic region of the plasma membrane in rod cells, and this gives rhodopsin its function as a transmembrane protein. There are a few polar amino acids that are important to the protein, such as threonine and glutamate. They allow rhodopsin to form a systolic tail, which aids in the G protein signaling cascade. And there are a few remaining amino acids that make up beta sheets and random coils throughout, but those aren't as structurally and they're not as relevant to the protein's actual function. The isoelectric point of the protein is 6.21. Rhodopsin is also known as visual purple, and as I mentioned, it functions as a G-coupled protein receptor, or G-protein coupled receptor, sorry. And rhodopsin is found in the retinal photoreceptors of the eyes. Structurally, it is composed of a protein opsin, which is covalently bound to a small light-sensitive molecule called retinol and 11-cis retinaldehyde. 11-cis retinaldehyde is a pigment molecule that is derived from vitamin A, and the protein overall is mainly expressed in the retina and is responsible for the rod's ability to see in low light. When a photon of light is absorbed by the ground state chromophore, a conformational change is elicited, and it starts a phototransduction cascade within the rod cells, which converts light into an electrical signal. The protein opsin dissociates from the retinol during this conformational change from cis to trans, and this causes bleaching. Bleaching is basically limiting the stimulation of rods, which allows for the cones to mediate vision, and it enables better vision overall, specifically in dim light, and this will allow animals to better survive in their environment and sense for food, mates, or threats more effectively. There are two main disorders associated with the protein rhodopsin: retinitis pigmentosa and autosomal dominant congenital stationary night blindness. There's no overall treatment for either of them, and this is mainly attributed to the fact that there are over 150 RHO gene mutations that cause these disorders. Basically, by altering the folding or transport of the rhodopsin protein contributes to this, because it interrupts the normal signaling that happens within the rod cells by mutations on the RHO gene. And as a consequence, the rod cells can't relay signals to the brain as effectively, and this leads to a loss of vision in low light. There are a few aids, as you can see here, that help somewhat mediate and crutch this, but like I said, there's no overall treatment. However, rhodopsin is a well-studied protein, so the outlook in the future looks promising. Medically speaking, advancements, there's potential. Thank you for listening to my presentation, and here are my references.